to my curious little children. I hope all of you are doing great, and let me just see if at all I am audible as well as visible. Just a quick second, okay? Where is it? Where is it? Yes, I hope it's perfectly fine. Sophia, it's 4:30. Later. Okay, it's fine. So, my dear children, we are here going to cover the second part of our chapter, that is sexual reproduction, flowering plants. In the first part, I remember we have completed the male reproductive part as a whole. Okay, each and every part of it has been done. So, make sure if you're someone who has not completed that or probably not watched that video, make sure you watch it such that your concepts have been cleared one by one. Okay, do not lose the track in between, because that would be not good. Hi Sophia, I hope you're doing great. Okay, so I hope we are ready for today's class, machas and machis out there. And today we are going to go ahead with megasporogenesis, as in we are going to talk about the female reproductive body of the plants, as well as we will talk about fertilization, double fertilization event. This is one of the most important topic out there, so make sure you are not missing out on this, and make sure if at all you have any doubts, ask me. I'm your friend. Okay, consider me as your friend and I'll help you out throughout. Okay, so ready? I hope we are ready. Let's go ahead. Sophia and everyone else who is watching this video, let's go ahead. I know that as of now you might do not have much mood to study, because some of you have not even started off the second PUC classes. Okay, in your class it might not be like that. So yes, Rizwan's question. What is Rizwan's question? Hi mom, I want to study when the syllabus will end. Okay, so Rizwan, the structure of it is made, okay, it is ongoing. Actually, we are not revealing the teachers because there is some restructuring that is happening depending upon the courses and things like that. Once it has been finalized, we'll surely let you know about that as well. Okay, and yes, your 11th and 12th, everything is going to be covered over here, so you do not need to worry about that. And it's not like you have to take up the Pariksha Veera series right now. If you wish, you can just wait for some time and check out the schedule whenever it will be released, and then also you can go ahead. But that might be a bit late. But yes, if you wish to have a proper schedule being seen, which I understand, you can duly wait for some time as well. Works? Okay, so right now we'll go ahead with the things that are going to be taught in this particular session, and the first part of this particular session would be what? The female reproductive organ. That is all about gynoecium. You know, the male reproductive part was known as androecium. Here also you have a separate name for it. What do you call this? Gynoecium. And if at all we dissect the gynoecium and see the exact structure, it looks something like this. So there is a stigma which is a highly sticky. Why sticky? Because the pollen grain which is there, okay, the pollen grain which is there, it has to land on the stigma over here, and to make sure that it stays here, it has to be sticky as well. So you have a stigma, you have a very long style, and finally you have something that is known as the ovary. So there is stigma, there is style, and there is ovary. Now depending on different type of flowers, the structure can change. It's not like the same structure will be followed, it can be different. So now I am going to get inside this and I am going to decode the structure of gynoecium. Gynoecium is also known as pistil, and sometimes to confuse you a little more, they also call it what? Carpel. So carpel, pistil, gynoecium is all the same thing. That is the female reproductive part of the plant. Cool. Now, as I said, we are going to see much more in detail how exactly it is, the structure it is, and I bet that this is definitely going to be highly useful for you guys. So, Baba, this is the stigma that I am talking about. Okay, here is the style and here is the ovary, and I'll just label it as well such that none of us forget it. Stigma, style, and ovary. Now, listen to me very carefully. But carpel and pistil, it's the same thing, Kavita. Carpel and pistil is the same thing. Now going ahead, over here when you talk about the ovary, we have to get inside the ovary and check what is in there. We have some very important part over here which you call as what? Ovule. Which you call as what? ovule. And ovule is the place where actually the female gamete will be found. The female gamete is found inside the ovule. Okay, and what do you call this ovule as? Do you have any other name for ovule? Ovule is also known as megasporangium. It is known as megasporangium. Cool? So I hope you remember in the case of males, there is anther. Inside the anther, you had microsporangium, right? Inside the microsporangium, what did you have? You have sporogenous tissue, and the sporogenous tissue was made up of what? The pollen mother cells, right? Similarly, over here, inside the ovule, Baba, which is also known as megasporangium, you have so many cells. You would be having so many cells arranged together. You say that these cells together form something like this, which is known as nucleus. And there is one very important cell right over here, which is going to be what? The megaspore mother cell. There we had pollen mother cell. Here we would be having the megaspore mother cell. Pollen mother cell was giving rise to pollen grains. Here the megaspore mother cell will be giving rise to megaspore, which will later form embryo sac. I know that will be too difficult for you to understand all of it together, so we'll go one by one. Okay? Aryan, hello. I hope you're doing good. Okay, so we are right over here, and I do not want to confuse you, which is why I'm going to remove certain things from here at this particular point. Okay, I'm going to remove certain things from here because that will be a lot of confusion for you guys. So as of now, we know ovary is there and ovule is there inside the ovary, and the ovule is also known as megasporangium. Now we are going to decode this particular part, because this is super important. So over here, I say this is the ovary and I have my ovule. Now ovule which is there, it cannot just stay in the air. It has to be connected to the ovary in some ways, right? And how do you think the connection is there? There is something like this right over here, which you call as placenta. Ma'am, why there is placenta? Placenta is something that is going to nourish this particular ovule. This ovule which is over here, it needs nourishment, and the placenta provides nourishment. Now obviously to connect both of them, you need something right over here. What is this something that you find right over here? You have a name for it. You call it what? Funicle. What do you call it? Funicle. And the point where it attaches to the ovule, you have another name for it. What is it known as? It is known as hilum. It is known as hilum. So you can just remember, placenta is saying hi to ovule with the help of the funicle. It helps in attachment of it. Okay. Now, ovule which is there, it feels that, oh my God, it's too cold over here. Can you give me a sweater? So basically, there are two sweaters. Okay, there are two sweaters. There is inner integument and there is outer integument. What is it? There is inner and outer integument. Outer and inner integument. So this is like a sweater, not as actually a sweater. It's basically a protective covering that we have around the ovule right over here. Why are we protecting it very much? Because this is a place, as I told you, where you will be finding the female gametes inside this. So this is a very important place and you need to safeguard it. Now over here, you might think that, ma'am, you have not buttoned the sweater. Why it is open over here? So this space is very much important for us. You might think, why? The reason being that I told you the female gamete is going to be formed over here. Now, if at all you want sexual reproduction to happen, male and the female gamete has to come together. From which point can they come together? Only through this particular point. So basically you have an opening over here such that the pollen grain which is there, when it made the pollen tube and when it is carrying along the male gametes, it can come inside through this and basically fuse with the female gamete. So there's a pore given over here, and this pore which is there, it is known as the micropyle. It is known as what? The micropyle. And this end where you have the micropyle, you call this region as what? Micropylar end. What do you call it? You call this as micropylar end. Okay. What about the opposite end when there is no micropyle? You have a name for that as well. The opposite end where you do not find the micropyle, name of that is the chalazal end. What do you call it? The chalazal end. Got it? Now I'm going to revise and you're going to listen to me. You have stigma, style and ovary. Together this is what? The gynoecium, or you call this as female reproductive part, or you call this as pistil or carpel. You have separate names for it. Now over here I am just zooming in the ovary and seeing what's in there. So in the ovary I have the ovule, which is attached over here with the help of a placenta. Now placenta and this has a connection in between, which you call as funicle, and the point where it attaches, it is known as hilum. Over here, the ovule is covered by two integuments, outer and inner integuments, and there's an opening in between. What do you call this? This is the micropyle. This is where the pollen tube is going to get entry in. Okay, enter in. Now over here, this particular place where you have the micropyle, where you have the pore, you call this as micropylar end. And the opposite end is known as what? Chelazal end. What do you call it? Chelazal end. Cool. Now, going ahead, inside the ovule, what will be there? Initially, you will not find the female gamete and things like that. Inside the ovule, there is one thing that is very important for us, and that would be what? The cells of the nucleus. So there are so many cells over here. So many cells are being lined up over here, and these cells are what? The cells of the nucleus. And in between that, we have a mighty cell in between. One cell, the queen of all. What do you call this as? You call this as MMC. Ma'am, MMC means a microspore mother cell. No. In the case of males, it is known as microspore mother cell. Here in this particular case, it is known as what? Megaspore mother cell. Megaspore mother cell. There it was microspore, here it is megaspore. This megaspore mother cell is the one that is going to undergo division and is going to form megaspore. This megaspore is going to form what? The female gametophyte. The female gametophyte as in the one that will later form the female gametes. That is known as the embryo sac. So just to give you an idea and such that you not get confused, let me tell you, let me make you understand over here, relate between the male and the female. In the case of male, you saw that you had what? Microsporangia. You had microsporangium, 4 in each corner. I mean 1 in each corner, 4 of them were there. Inside that, the innermost thing that you had over here was sporogenous tissue. What was it? Sporogenous tissue. Here you saw that all of the cells which were there, they were what? Microspore mother cell. They were all microspore mother cell. They were all dividing. They were undergoing meiosis 1, giving rise to 2 cells. They underwent meiosis 2. At the end, they gave 4 cells, that is 4 microspores. And what happened to these microspores? All of them basically gave rise to pollen grains. So this gave rise to pollen grain, this rise to pollen grains, this gave rise to pollen grain, this also gave rise to pollen grains. And each pollen grain over here is known as gametophyte. Why? Because each of them, okay, inside them, what do they have? They have the gametes. So they are gametophyte. Why? Because they will be forming gametes later on. Now over here, when I compare it with female, you need to understand one very important thing. That is, here the role is twisted, a bit twisted. You have ovule. This is what? Ovule. And ovule here is known as what? The megasporangium. It is known as megasporangium. There the microsporangium was microsporangium. You didn't have any other name. Here the ovule is known as megasporangium. Yeah. Inside this, what did you have? You have sporogenous tissue. Here what do you have? Here you have cells of the nucleus. Here you have cells of the nucleus, and here you have one very important cell which is there. Only one very important cell. Here inside the the sporogenous tissue, you have multiple microspore mother cell, but here inside the nucleus you just have one. Okay, you just have one megaspore mother cell. Only one, not all of them are going to be like them. Okay, here only one guy is there, and that is the megaspore mother cell. This megaspore mother cell is going to undergo meiosis, is going to give rise to 2 cells. Further, this will again divide, give rise to 2 cells at the end. right? They are all known as what? Here they are known as microspores. Here they are known as what? Megaspores. Here they are known as megaspores. Cool. Now all of them, do you think they will be giving rise to the female gametophyte? So here all of them were giving rise to the male gametophyte, that is known as the pollen grain. Here always remember, only one gives rise to the gametophyte, and that gametophyte's name is what? Embryo sac. Embryo sac. So ma'am, what happens to the three of them? Three of them degenerate, only one forms the embryo sac, and within the embryo sac, Baba, you will be having, inside it you will be having the gametes being formed. Inside it you will be seeing that the gametes are being formed. So over here all the cells were giving rise to what? Multiple pollen grains. Each one cell gave rise to 4 pollen grains. That is how it worked. But here you just have one megaspore mother cell. In the one megaspore mother cell, only one is going to go ahead with embryo sac formation, and basically that will be having the gamete. So basically, can I say, inside the ovule, later on, okay, later on, once division and everything has taken place, you will be having an embryo sac over here. You will be having what? An embryo sac over here, and the embryo sac will be having gamete inside this. That is the logic behind it. Now what happens to the cells of the nucleus? That is not participating, right? So usually the nucleus is there to nourish this particular cell. Okay, sometimes it can stay, sometimes it can be fully utilized as well. So that means even after formation of embryo sac, sometimes it stays. Okay, or else there are chances that it can be fully utilized and it will be just embryo sac rather over here. That is how it usually goes. Cool? I hope that works. Now, Shalini, if I start now for NEET, cannot guarantee you about the score, but I'm pretty sure if at all you start working hard, you can really boost up the score from the previous score that you have got. So you had got 229, right? So you can increase your score if at all you give your best, because the examination is on 21st June. We still have time. Give your best. Cool? Okay, so this is what I was talking about. I hope you guys are understanding. I know it's a bit difficult, but slowly you'll get a hang of it. So here you have microsporangium, there you have ovule as megasporangium, here you have sporogenous tissue, here you just have cells of the nucleus out there. Here all the cells of sporogenous tissue are going to give rise to microspore mother cells. Here only one cell is there. Among this, only one cell is there, which is known as megaspore mother cell. Here this microspore mother cell gives rise to 4 microspores. Here also they give rise to 4 megaspores. Here all of them give rise to the male gametophyte, that is the pollen grain. Here only one develops into the female gametophyte, that is known as embryo sac. You get it? So things are quite different here and there. Cool. Just have the comparison learned and it will be really helpful for you. So we studied about the structure of this, and now we're going to read about this so that everything gets cemented in your brain. So ovule, which is known as megasporangia, arises from the placenta inside the ovary. We studied about it. Ovule, which is there, arises from the placenta inside the ovary. So this is the ovary. This is basically the cavity of ovary. What do you call the cavity? This is the cavity where the ovule is located, right? You call this as locule. The cavity which is there, you call this as what? Locule. Okay, I hope that works. Yes. Now, placenta is located inside the ovarian cavity. We just studied it. Inside the ovarian cavity, you have the placenta. Okay, ma'am. Funicle, stalk attaching ovule to placenta. Stalk attaching what? Ovule to placenta, isn't it? Yes, that is funicle. That also we know, ma'am. Next, hilum, junction between funicle and the ovule body. This junction which you have between the funicle and the ovule is known as what? Hilum. Yes, ma'am. Yes, ma'am. Integuments, 1 or 2 protective layers leaving a small opening, that is known as micropyle. So over here, integuments, you have 2 integuments. There are 2 layers, and over here you have a small opening, and this is known as what? Micropyle. Cool? Yes, ma'am. Now, chalaza, opposite end of the micropyle, base of the ovule. So the opposite end of the micropyle, this is the micropyle, the opposite end will be known as what? Chalazal end out there. And finally, what do you have? Nucleus, mass of cells with reserve food. So over here, it is filled with so many cells out there. These cells are basically a part of nucleus, and basically they are there for providing nourishment to the MMC, that is the megaspore mother cell. That is the basic thing about this. Is there any doubt that you guys want to ask me? If yes, please ask me right away, because that is how it is going to be fun. Otherwise, it's just me teaching, and it will be very boring for you guys. If you have doubts, don't think that what will ma'am think if I ask a doubt like that and things like that. Listen, you will have doubt only if you study, right? So, yes. Sabha, ma'am, your Master Stroke series helped me a lot in KCET and NEET 2026. Is it? Oh my God, that's so sweet of you. Thank you so much, Sabha. It means a lot. It feels so good to see you guys coming back and commenting, and I'm really, really happy for you. I hope your examination went well for KCET and for NEET. I hope you are giving your best try for the next examination as well. Yeah? So cute, so cute. Thank you so much. Okay. So yeah, so I hope this part of the story is clear. I'm pretty sure that not very much people are watching this video at this particular point. You might not be serious about KCET at this particular point, but whenever you watch it, you should understand it. That's why I'm just confirming that. Yes, yes, okay. So that's the part of it, and now I also want to add one more thing. So we saw that this is the ovule which is there. Now you have some other name, okay, a technical term that you need to know, and that is anatropous ovule. Anatropous ovule, because this is also given in your NCERT. What do you mean by anatropous ovule? Anatropous ovule is where it becomes twisted. Okay, the ovule is kind of a bit twisted, but it's not twisted. Okay, it's not actually twisted. Just listen to me. It's very easy. The placenta and the funicle, which are right over here, it's now shifted over here. Just think like that. For making you understand the picture I'm telling you, the placenta and the funicle right over here is going towards this side. So basically, if at all I have to reimagine, it will be removed from here and it will be right there. The placenta will be here and the funicle will be here, and the point of attachment will be known as hilum. So the same picture is given right over here to confuse you guys. This is known as the anatropous ovule. You have the the embryo sac that is within the ovule. I told you will be having embryo sac and the nucleus, inner integument, outer integument. Over here you have the micropyle, and just near it you have what? Hilum. Hilum is the place of attachment. Attachment of what? The funicle. And here you will also be having placenta, which is not shown over here, but it will be there. Cool. I hope that works. So this is one very important thing. Make sure you go through it, and if you have doubts later on while you watch this video, you can ask me also. If you do not have doubts, if you think it's very much understood, let me know about that also. Cool? Yes. So let's go ahead and talk about the next thing which is there, and that is the types of pistils. So listen, the carpels or the pistils. So over here there are conditions where a flower will be having just one carpel. That means only one stigma style ovary. That is known as monocarpellary condition. What do you call it? Monocarp. Mono means one. Okay, mono means one. That means one carpel is there. Cool. There are chances that a plant or a flower can have multicarpellary condition. Multicarpellary means multiple pistils are there. So many stigma style ovary, stigma style ovary, stigma style ovary. You get it? That is known as multiple condition or multicarpellary condition. Now in multicarpellary condition, whenever you have more than one, there is problem in there. There can be problem in there. So the problem is, it can happen in two ways. One is, it can be syncarpous, and the other one is apocarpous. Ma'am, already botany is killing me with all the terms. I do not want more terms, but I cannot help you over here because we need to learn about this. Listen to me. Syncarpous condition means, okay, it's very simple. Just imagine. You have stigma style, stigma style, stigma style, but the ovary is fused. What is happening? The ovary is basically fused. So multiple stigma and style you can see, but the ovary is fused. Since there are many of them, the ovary is fused. This is known as syncarpous condition. What is it? Syncarpous condition. Now on the other hand, you have apocarpous. Apocarpous means all of them say, Macha, you stay away, I will have my own ovary, you also have your own ovary. So stigma style ovary, multiple of them, and they are not attached to each other, they are free. That is known as apocarpous condition. And when I show you the picture, it will become all the more clear, and you will be like, oh my God, this is the picture that is given in NCERT. So listen to me. Syncarpous condition is shown with the help of a picture like this. Okay, now it's actually funny that they have given this picture, but yes. So syncarpous ovary is mentioned over here, stigma is mentioned over here. Now how do you expect a child to understand with the help of this picture? I have no idea, which is why I have taken a picture of the same plant that they are talking about over here, and I am trying to show you over here. Listen, All of them have the stigma, they have the style also. Do you see the ovary? Ovary is obviously going to be inside. Well, you do not see the ovary. Why? Because the ovary is fused. It is going to be what? Fused. Always understand that. Sorry, it's not the stigma and style. Basically, they have the stigma and the style over here, and it's all separate, but the ovary over here is going to be fused. What is it going to be like? It is going to be like fused. Always remember this. This is very, 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 very important. I hope that is clear. So this would be the stigma, the sticky part of it, most probably, and then below that you would be having everything else. Cool? The plant which is having something like this, what do you call this plant as? This is basically papaware. This is papaware plant. This is where you get the drug opium poppy from. Okay, you get the drug opium poppy from this particular, or basically this particular plant is known as opium poppy, and from here only you get the opioids or opiates. So please know that. So you have papaware over here. That is the example of syncarpus condition. Now you have apocarpus condition as well. What do you mean by apocarpus condition? Apocarpus mean, as I told you, the stigma and the style and the ovary are separate. Now you might have seen this particular plant. Now the picture in your NCERT is given something like this, where they have mentioned carpels are there, and you have to understand from this that the carpels are separate. Now, the actual picture, the actual plant that they're talking about is this. You might have seen this particular plant. You call it champak in Hindi, chembagam in Malayalam. It smells so good. Okay, and if at all you just move aside the petals out there, you will be seeing something like this, and this is the picture that they have given for it, where the stigma and the style are separate, and also the ovary will also be separate. This is apocarpus condition, and this plant that you're talking about, which I say it's known as champak and all, the actual name for it, the scientific name for it will be what? Michelia. It will be Michelia. So please remember it. So you have papaware for syncarpus condition, you have Michelia for apocarpus condition. This is very, very important out there. And so sorry for saying it's a stigma and the style. You have the stigma over here. Okay? Cool? Works? Understanding things? Learning something new? Or is it like the same things that we are learning? Okay, and trust me, this channel might, this chapter might feel like boring for you at the start. Okay, it is actually a little bit boring, but you will get a hold of it. You will find it interesting later on. Now, just give me a quick second. Okay, just a second. Yes. I just want to make sure. Okay. Just a second. Okay. Cool. So we're right over here. I hope that is clear. Yes. So sorry, I had an important message to be put, okay, in the official group. So yes, this is how it works, and I hope everything is clear. So and also, by the way, this is the stamen. Okay, this is the stamen, and this part will be the stigma. I'm recorrecting again such that you do not know any mistakes. Okay, fine. I hope this is also clear, and now we move forward. Please remember the names of them. This is important. This is multicarpally condition. Now there are other cases where you find multiple ovules. So as of now we were just talking about multiple carpels and things like that. What if inside the ovary there are multiple ovules? Is that possible? Obviously yes. And where do you see that? So for that you need to understand the logic. The logic is, whenever fertilization happens, you have the ovary, right? Ovary becomes the fruit. What happens to the ovary? Ovary which is there, it will turn out to be the fruit. What about the ovule? The ovule which is there, it will turn out to be the seed. It will be turning out to the seed. So suppose you will be having multiple ovule, that means that you will be having multiple seeds. Now what are those plants where you are going to find multiple seeds? Papaya. In papaya, you've seen multiple seeds are there, right? Watermelon, you come across multiple seeds. So that means that these plants will be having multiple ovules. Now, the things that can be difficult for you to remember would be orchid, because this is something that we generally do not eat, right? So orchid is also there coming under a multiple ovule condition. So please remember that. Now, on the other hand, when you talk about wheat, paddy or mango in general, all of it, they just have one seed. In mango, there is only one seed, right? So that means single ovule is there, because that ovule is only transferring or converting to a seed, right? So that means they just have a single ovule. Similarly, wheat and paddy, everything, they only have a single ovule. That means a single seed is there. That is the thing that we eat. You get it? I hope that's sorted. So please remember, it's a very easy thing, but I just wanted you to know about it. Cool. So that is also done. And now we go forward and about Pariksha, Veera. This is one very important thing before I go ahead, and that is, my dear children, those of you who are watching this video, if at all you think that this video is helping you well, please understand that you will be getting multiple videos like this by multiple other teachers who will be taking care of physics, chemistry, mathematics and also biology in the Pariksha Veera batch, which is there. It's a KCET 1-year online program and is supposed to start by the month of June, by June 15th. And the pricing for that is rupees 3,499. There are live classes that is going to be there. There will be recorded classes. Your class PU1 and PU2, everything is going to be completed. You will be getting daily practice papers containing at least a few questions out there, so that you can also practice, and all of it will be done for sure. So if at all you want to know more about it, you can check out on the link that is given in the description section. Please make sure you do that. That will definitely help you out. Cool? Yes. And now we go to the next thing that is known as Megasporogenesis. As I was talking about it already, I spoke about it already. Remember, I told you that megaspore mother cell divides itself. We're going to see what is going to happen in real life. Okay, so let's decode that. So over here, when I was talking about the ovule, I told you you have so many cells that constitutes what? The cells of the nucleus. Among that, you just have one cell which is pretty much like a queen out there, and this cell is known as what? The megaspore mother cell. Only one cell is there. This megaspore has to undergo what? Division. What division? Meiosis. Always remember, from this particular point of time, it's preparing itself to form the gametes later on. It is not forming the gametes directly, it is preparing itself. So we have to talk about meiosis. Why? Why meiosis? I have told you in the previous video also. At the end of meiosis, you see that if at all the number of chromosomes were 2n, it will become half, it will become n. And why do you think it's important over here? The reason being that, listen to me, one gamete is having half the number of chromosome, another gamete is having half the number of chromosome. N plus n, it will become 2n. That means it will be turning out to be like a normal cell of a body. But suppose there is no reductional division, there is no reduction happening over here, it is 2n. So in the male gamete will bring 2n, the female gamete will bring 2n, it becomes what? 4n. It will be double of what a normal cell has, right? And that is not fair. That's exactly why we have meiosis being undergoing over here. So you have the megaspore mother cell, which is this. It undergoes meiosis 1. Meiosis happens in 2 stages. It undergoes meiosis 1. Suppose it was 2n in nature. Over here, when it undergoes the first round of meiosis, it becomes reduced to half. The number of chromosomes goes half over here. Half of them will be going in the other cell out there. What happens next? Again, meiosis 2 is going to happen, where this cell is going to divide into 2 and this cell is also going to divide into 2. Now here you need to pay attention. This cell which divides into 2, do you think the number of chromosome again become half because it is meiosis? No. Meiosis 2, which is there, here the number of chromosome remains the same. It does not get half. What happens? It remains the same. Always remember it. This is very, very, very important. Okay? In meiosis 2, it remains the same. Now, these 4 cells that you have over here, what do you call them? What do you call them? So, first of all, what do you call them? You call them as megaspores. And this process is known as what? Megasporogenesis, because you are forming what? Megaspores. Now, there we saw that all 4 of the microspores which are formed, they give rise to the pollen grains, which are the male gametophyte. But here, things are going to be different. 3 of the megaspores are going to what? Degenerate. Chum. Okay, what about the one? Only one remains functional, and that is known as the functional megaspore. What do you call it? Functional megaspore. What does it form later on? Basically, it forms the embryo sac later on. It forms what? The embryo sac later on. Ma'am, what is embryo sac? I do not know what is embryo sac. It is basically the female gametophyte. Please remember this name. It's not the female gamete. It is the one that forms the female gamete. Just like pollen grain was the male gametophyte, it forms the male gamete, right? Similarly, embryo sac is the one that is going to later form the female gamete out there. So always remember that. So what have we just studied? We studied what is megasporogenesis. It is nothing but the formation of megaspore from the megaspore mother cell. Then we studied about MMC location. Did we study that? We did not. So we have to study that. Megaspore mother cell, where do you think is located? So we saw that there is a very big ovule over here. Okay, very big ovule over here. Where is the megaspore mother cell located? It is located towards the micropilar end, not towards chalazial end. It is mostly located towards the micropilar end. Always remember MM. Okay, in that way you can remember it better. Cool. Now that portion is important because you will get questions related to it. Now the next thing that is there is MMC division, that is this division. It undergoes meiosis producing what? 4 megaspores, okay, but only 1 megaspore remains functional. And what do you call them? The functional megaspore. What happens to the rest 3? Rest 3 of them, they degenerate. Now listen to me. I told you that 1 megaspore which was there, it forms the embryo sac, which is why you will call this development as what? Monosporic development. Monosporic means only one spore develops further. Monosporic development. This is very important. They'll ask you why it is known as monosporic development. So you have to tell, Baba, there were 4 of them, but one was the only one functional, rest all of them degenerated. Only one is responsible for the formation of the female gametophyte, that is the embryo sac. That is why. Now you might think that, ma'am, you're talking about this question, but I'm pretty sure these type of questions do not come in KCET examination. If at all you have thought like that, then that will be wrong, because KCET had a question like this. So embryo sac is termed as monosporic development because of what reason? Why it is known as monosporic development? Because the development occurs from 2 functional megaspores. Is it why? It should be disporic, then it's not monosporic if it's 2 functional megaspore, right? Next thing, their development occurs from 3 functional megaspore. No. Their development occurs from 1 functional megaspore. Yes, that is the reason why it is known as monosporic. From megaspore mother cell, 4 linear tetrad megaspores are formed, out of which only 1 megaspore is functional. That could be more correct, right? That could be more correct. Or we have more reasons. That is what? Their development occurs from all the functional megaspores. That would be wrong, because that is not correct. So we have option this and this. And we are kind of very much worried what will be the answer. So let's reread it. From megaspore mother cell, 4 linear tetrad megaspores are formed, out of which only 1 megaspore is functional. Is it because of which you call it monosporic development? No. Listen to me. Yes, 1 megaspore remains functional, that is part of it, but that one is the one that forms the embryo sac, right? And that is why it is known as monosporic development. Being functional is different, okay? Forming the embryo sac later on is a different part. So over here, it's clearly mentioned that their development occurs from only 1 functional megaspore. Here they have told how the functional megaspore is being formed. That is not the question. So which is why the answer for this question is going to be what? C. It is going to be C. Now they can twist and give you questions like this for easier concepts, which is why I'm telling you, make sure you try to understand the concepts as nice as possible. Okay? Got it? So everyone who is watching this video, are you understanding things, or is it like shim, 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 shim? It will be really good if at all you talk to me in the chat session. I know that this is not the time where every one of us gets serious about KCET. I know it's so for some of you the classes might have not even started, but there is a reason why we are making videos like this right now, because we want to instill in you a little bit spirit about how good it feels to study early. Start studying early and probably rest later on. You will not rest later on. That will be not so honest thing to say. You will not rest later on, but you will have enough time to prioritize things that you actually want. You get it? You can do more things. Is it clear, children? If yes, can you give me a thumbs up? if everything is sorted, you are understanding things. Can you give me a thumbs up over here? Or are we too shy to give a thumbs up? I understand though, but I want you to slowly open up a bit, otherwise it will be a very boring class where Jasmine Ma'am is the only one who is talking. Okay? Okay, my children are not in a mood to talk, so I have to go ahead. So I hope this part of the story is clear. Now, when I say that it forms the embryo sac, it's not a one-step process. So it just had underwent meiosis until now because it want to reduce the number of chromosomes out there. Now things are gonna change a bit and you need to know about this. And we are gonna see how exactly will this happen. That is, how exactly is embryo sac going to be formed, or what do you call as the female gametophyte? How do you think it is going to be formed? Let's check it out. So over here, okay, over here, the first thing is that there is this cell that is your functional megaspore. What is this? This is your functional megaspore which is there. Cool. This functional megaspore which is there will be having its own nucleus. Yes. What type of nucleus? This nucleus is going to be haploid because it has underwent what? Meiosis. That's why it is going to be haploid in nature. Okay. Now our task is to form an embryo sac. An embryo sac is a very big structure with multiple cells, but over here we just have one cell. So what is going to take place? Mitosis is going to happen. Mitosis is a process by which you can form multiple cells with the same number of chromosomes. It's different from meiosis. In meiosis, reduction happens, but in mitosis, the number of chromosomes remains same. So for that, before the actual process begins up, there is something else that happens. The nucleus which is there, it is going to divide. How many times? It is going to divide thrice, and you need to remember it. It divides thrice. I'll say that basically, okay, basically over here, I'll say they undergo division thrice, or I'll say they undergo 3 mitotic division. Please remember this. This is very, very important. How many division? 3 mitotic division. Very important. Okay? Yes. Now see. So when I say there is a nucleus over here, it divides into 2. Ta-da! You have got 2 nucleus. Now again, the number of chromosome remains the same. It is just N and N. Cool? Now further, what is going to happen? Let's see. Okay, just a second. Let's see. So over here, again it is going to divide. This cell, this nucleus which is not cell, the nucleus which is there, it's again going to divide over here N and N. Here also it becomes N and N. Cool. Further, what happens? One more division happens. Here it becomes N and N. Here also this divides into 2 nucleus N and N. Here also divides into 2 nucleus N and N, and finally N and N. So 3 mitotic division happens, but the end of it, how many nucleus do you get? You get 8 nuclei. If at all you count 1, 2, 3, 4, 5, 6, 7, 8. You have got 8 nuclei. You have got 8 nuclei. This is very, very, very important thing. Cool. I hope you guys are understanding it. Yes. And ma'am, I know I can't get here. Ayyo, Karthik, that's so cute of you. Okay, so anyways, so there are 8 nuclei out there, and over here we need to arrange this nuclei as well. How will we arrange this nuclei? Let's see. So my dear children, before I go ahead with that, one more very important thing. We just spoke about nuclear division. What happens to the cell? Cell division does not take place. So actually the nuclear division that takes place over here, we say it is what? When we say nuclear division is taking place over here, we say it's free nuclear division. What do we say? We say it's a free nuclear division. This is one very, very, very important thing. Okay, that means that it is not going to form cell wall around it immediately. Later on, when 8 nuclei are being formed, slowly cell wall are going to be around this particular thing, making it into different cells. And here comes a tragedy. Tragedy is not the correct word to use. Here comes a, what do you say, surprise to you guys, and that is this. So when I say that the division is happening over here, and when I say that the free nuclear division is happening and later on it is forming the cells and things like that, you need to understand one very important thing. How many cells do you expect to form? You expect 8 cells to be formed, 8 nuclei are there, 8 of them, if at all they're surrounded by cell wall, it should be what? 8 cell at the end. But over here there is a twist. And what is a twist? Twist goes like this. 3 of the cells are being formed with nucleus. You call them as antipodal cells. What do you call them? Antipodal cells. Now again, 2 cells are being formed over here. They are known as what? Synergid cells. They also have nucleus. This is known as synergid cell. One more cell is being formed over here. This is your egg cell. Egg cell means it is going to be what? Your female gamete. This is the one that is going to be known as what? The female gamete. Now what happens? How many cells are being formed? 1, 2, 3, 4, 5, 6. 6 of them are being formed, ma'am. How many do we want? We want 8 cells. Do you think we'll get 8 cells? No. We'll only get 7 cells. How, ma'am? So this particular portion that you see entirely over here, this is another big cell which is there. What do you call this cell as? This is the central cell. But ma'am, we had 2 more nuclei. What happens to the 2 nuclei? The 2 nuclei right over here will be occupied by this big cell. That means it will be taken up by the big cell. So the big cell has 2 nuclei? Yes. So you will consider it only as 1 cell? Yes. You get it? The cell has 2 nuclei, please remember it, and these 2 nuclei are known as 2 polar nuclei. They are known as 2 polar nuclei and they belong to the central cell. So in that particular condition, if at all I ask you, how many cells are there in an embryo sac? Now this structure is known as an embryo sac. How many cells are there in an embryo sac? You will say, ma'am, 1, 2, 3, 4, 5, 6 and 7. 7 cells are there in an embryo sac. Okay, Baba, how many nuclei are there in an embryo sac? 1, 2, 3, 4, 5, 6, 7, 8 nuclei are there in an embryo sac. So you will say, it is 8 nucleate but 7 celled in condition. It is 8 nucleate but 7 celled in condition. So you can say 8 nucleate and 7 cell as well. That is the ulta of this. Is either way, it's the same thing. Please know that is how your embryo sac is being formed. This is very, very important. Your only 1 cell, that is the female gamete, is there, and that is known as egg cell. To guard them, you have 2 synergids. These synergids are like best friends of the egg cell. Okay, they are known as the best friends of the egg cell. Over here, the whole thing that is being formed over here, I say it is the egg apparatus. What do you call it? It is known as the egg apparatus. Why egg apparatus? Because you have the egg and you have the 2 synergids. Got it? This is important. Make sure you go through it. Very, very, very important. Cool. I hope you guys are understanding. I hope you guys are listening. Okay. So I'll just revise. Listen to me very carefully. You had 1 megaspore. The megaspore had 1 nucleus. This nucleus which is there undergoes 3 mitotic division. But I say it's free nuclear division. I have to write it. You might forget it. It is what? Free nuclear. That means immediately when the nuclear division takes place, it is not surrounded by cell wall. It's slowly surrounded by cell wall. Slowly. Okay. So initially it divides thrice. 3 mitotic division happens. It is covered by the cell wall out there. Okay, and that's how you get certain cells. How many cells? You expect 8 cells to be formed, but that's not it. There are only 7 cells which are being formed. 1 big cell is there which has 2 nuclei, that is known as 2 polar nuclei, and this very big cell which is there, that is known as central cell. Over here you have the female gamete which is guarded by 2 synergid cells, which is why you call them as, since they are always together, you call them as egg apparatus. Okay, what do you call it? Egg apparatus. Also one more very important thing, the synergid which is there over here, it is having finger-like projections like this. Okay, what do you call this as? The finger-like projections that you see over here, what do you call this as? This is known as filiform apparatus. Filiform apparatus. And why do you think filiform apparatus is important? I'll tell you the story. Before that, you need to just remember everything in your head and also know one very important thing. We were talking about ovule. Ovule has a micropylar end and a chalazial end. Remember, right? So over here, micropylar is the area where there is an opening. Opening should be ideally over here. Why? Because the female gamete is over here. It should be coming and fusing, right? So this place where you have the synergids, where you have the egg cell, we say this is where you have the micropylar end. This is where you have the micropylar end. Whereas the antipodal cells, when they are lying, it is known as the chalazial end. It is known as the chalazial end. Remember that. Okay, so whatever waste happens, it usually happens in the chalazial end. Antipodal cells did not have any specific function that is given in your NCERT, but you just need to remember it. It could be helpful for you. Cool. I hope that works. Yes. Take some time, and that's how we go and understand things. And as we were talking, over here, all the cells, since they were forming from the mitotic division, it will be having the same number of chromosomes, that is n number of chromosomes. So when I say that what is the ploidy of these cells, when you are being asked what is the ploidy of these cells, understand this is going to be haploid. Okay, this is also going to be haploid. This is also going to be haploid. That is, it has half the number of chromosomes only. Same like this. Similarly, egg cell is also going to be haploid. Synergids are also going to be haploid. What about the central cell? This is where most of you get confused in. What about the central cell? Central cell has two nuclei. One nuclei has haploid, just like everyone else. Similarly, this also is haploid, just like everyone else. Haploid, it becomes what? Diploid in nature. Central cell will be diploid in nature. Always remember, this is the only one which is going to be different. Rest everyone is going to be haploid. This is the only one which is going to be diploid in nature, and you should not be missing it by any chance. Cool? Yes? Understanding things? Children, are you listening? If at all there is anyone who have any doubt that you're coming across, you can ask me right away. If you have no doubts, we can go ahead as well. I think we can go ahead. Okay, so this is about the female gametophyte, where your gametes are going to be formed. And if at all you do not have the structural understanding of it, this is exactly how it looks. So as I said, you have the antipodals, you have the polar nuclei over here, you have the central cell, you have the egg cell, you have the synergids, you have the filiform apparatus. Everything is right over here, and this is very, very important for you guys to know about it. Cool? And if you are worried about the placement, it looks something like this. So over here, when I say that, okay, when I say that this is the ovule, here you would be having the MMC has later divided and formed the embryo sac, right? So here you would be having your embryo sac, and here you would be having your egg cell as well as synergids and everything. Wherever the micropyle is there you will be having your egg cell as well as synergids and things like that. And the opposite end, you will be having your antipodal cells. Okay, I'll just write it also. Okay. And now finally, we are going to go ahead and talk about the next thing which is there, which is what? The question. And then we'll go ahead with the next topic, which is double fertilization. Post that, we can end up the class. Cool? Okay. So given below is a diagrammatic sketch of an anatropous ovule. Anatropous means, I told you it's twisted, it's ulta, right? Select the correct set of names of parts labeled as A, B, C and D. Now this is also a KCET question, and we need to know about this. So listen, this is the picture that is given to you. What is A, what is B, what is D and what is C? That is the question that is being asked. So the easiest one to find out, okay, the easiest one to find out would be this, D1. Innermost one, it has to be an embryo sac. Innermost one, D1, it has to be an embryo sac. And here we're going to start using elimination method. Let's start using an elimination method. Embryo sac, so this option and this option goes off. You just have two things which is having embryo sac. Now with the help of this, you try to find the answer. Now funicle is same, that means A is going to be funicle. A is something that is probably stalk-like structure which is attaching over here. In anatropous ovule, you saw that funicle was here, hilum was also here, right? So what will be this? What will be B over here, if at all you are asked, or what will be C? C will be the opposite end of micropyle. If this is where you have the opening, the opposite will be known as what? Chalaza end. So C will be what? Chalaza. Right, it will be chalaza. And then what you are left with? You are left with B. What is B over here? Remember my dear children, you have the ovule. Inside that you have the embryo sac. Surrounding it you have the cells of the nucleus which can be used up or which can be still be there. So B is going to be the cells of the nucleus. So this and this is same, only this is different. With the help of it you can find the answer. What is the answer for this question? It is going to be C. It is going to be C. Okay? Cool? Done? Now we go ahead and we talk about the double fertilization event as well. So pollen pistil interaction as well as double fertilization is what I'm going to teach next. So make sure you listen to me carefully. Pollen pistil interaction, as in, I told you that over here pistil is what? The female reproductive part. This is the female reproductive part. You have the pollen over here which is going to land on the stigma, right? This is the stigma which is the part of the pistil. It is the part of the pistil. Now when I say pollen grain lands on it, I already told you pollen tube is going to form a pollen grain is going to form what? A tube-like structure. Why? Because it has to go inside the ovary, because inside the ovary you have the ovule. Inside the ovule you will be having the female gamete, right? So this has to form a tube-like structure and that is known as pollen tube. It is known as pollen tube. But do you think all the pollen which lands on the stigma is going to give rise to the pollen tube? The answer would be no. It would be no. This is very important. So over here, when I say this particular thing, always understand, here the chemical coordination has to be really good. Otherwise what is going to happen, even if the pollen lands on it, it will not be able to make the pollen tube out there. So there is recognition of pollen by the pistil, and either it can promote or it can inhibit as well. Two things can be possible. Either it can promote or it can inhibit. Now suppose it promotes, I will say that this process is known as germination of pollen grain. What do I call it? Germination of pollen grain. On compatible pollen pistil interaction, that means on in the case where the interaction is totally normal, it's totally cool, okay, where the interaction is happening positively, okay, it has recognized the pollen, it is promoting, I'll say that it is known as compatible pollen pistil interaction, and this will lead to germination of the pollen on the stigma, and it means that it is going to give rise to the pollen tube over here. It is forming the pollen tube over here, right? And when I say it forms the pollen tube, you need to understand that pollen grain which came over here, it can be of two ways, it can be in two conditions. One is that basically it has two cells, one generative and one vegetative, okay, one vegetative and one generative. This, if at all it comes like this, basically while forming the pollen tube, this divides into two, the generative divides into two gametes, Otherwise rest of the 40% of the pollen grains land in a 3-cell stage where they already form the two gametes and they have one vegetative cell. The vegetative cell over here is the one that helps in the cytoplasm of the pollen tube formation. The two gametes which are there, they are going to just move around. So please know that if at all they are coming in 3-cell stage, the male gametes just move around. If at all it is in a 2-cell stage, the male gametes are going to be formed while the pollen tube formation is happening, while the pollen tube is growing. So in plants where pollen is shed at 2-cell stage, the generative cell divides during tube growth to form the two male gametes. So while the while it is being formed, that is the case where the two male gametes are going to be also formed, while the pollen tube forms, okay. Now the pollen already contains, okay, that is okay, in plants with 3-cell stage, I see 3-cell pollen, I already told you they already have the two male gametes, so it will be going as such itself, okay. Now lastly, entry into the ovule. The pollen tube enters into the ovule through the micropylar end. This is important. I told you that when it enters, it is going to enter through the micropylar. Where is the micropylar end? This is the micropylar end. Wherever you have the egg cell, that is a micropylar end. And I told you the pollen tube is going to be formed through here. Okay, it is going to enter, not formed, it is going to enter through here. When I say it is entering through here, who guides the pollen tube? That Macha, you have to come over here. It's the synergids. Okay, the cells, the synergid cell over here, I told you, it has something like this, which is known as filiform apparatus. The filiform apparatus of over here says that pollen tube, you are carrying two male gametes, right? You're carrying two male gametes. You have to come through this particular route. I will guide you. Who is that? Filiform apparatus of the synergid cells. So, ma'am, if at all that happens, don't you think the synergids will die? Yes, synergids are gonna die. Whenever the pollen tube formation happens through this, the synergids are going to die, degenerate in this particular process. Not both of them, at least one of them might degenerate. But ma'am, I have one more doubt. Okay, you told that filiform apparatus is there to guide the pollen tube and things like that. It's totally fine. Agreed. But why there are two male gametes when there is only one female gamete? What is the reason for that? That is another question. And here comes the role of what? Double fertilization. Here comes the role of double fertilization, which you should absolutely not miss. So listen to me carefully. If you hear me out carefully, you will not forget anything that is going to be coming from this particular portion. So my dear children, over here what do you have? You have the three antipodal cells, agreed? All of them haploid, haploid, haploid. Here you have what? The synergids, which are also haploid in nature. Here you have your female gamete, which is also haploid in nature. So I'm just writing it. Female gamete. Okay. Then you have what? You have one more cell, that is the central cell. This big space that you see is all the central cell. The central cell will also have two nuclei. It is what? Having two nuclei. What do you call this nuclei as? You call this as polar nuclei. How many nuclei is there? Two of them. Ma'am, in one cell, yes. What do you call this big cell as? You call this cell as central cell. I already told you. Now listen to me. So there are two male gametes, okay? The first male gamete, I'll call it Chunnu. Okay, I'll call it Chunnu. Chunnu and Munnu. So Chunnu male gamete, the first male gamete over here, which is also haploid in nature, by the way, it is also haploid in nature. It comes and fuses with the female gamete over here, because that's the usual thing that is to be done, right? It comes and fuses with the female gamete. Oh my God, then what happens? It forms what? Zygote. It forms the zygote. So after sexual reproduction, we always see zygote is being formed. Here also that happens. Okay, ma'am, so you are saying female gamete, which was haploid, and the male gamete, which was also haploid, comes together to form zygote, which is going to be diploid? Yes, Baba, that is the total logic. It has to be diploid just like a normal cell. So it is diploid. Okay, what do you call this as? Fertilization. What fertilization? What do you have the exact term for it? You will call this process as syngamy. What do you call it? You call this as syngamy. This is the first fertilization that happens, and this is known as syngamy. Very, very, very important. Okay, now we go ahead. Ma'am, one more male gamete is there. What happens to that? Let's see it. So one more male gamete which is there, it is also haploid, as in the cell is there, it will have a nucleus which is haploid in nature. This comes and basically joins up with the two polar nuclei of the central cell. That is a twist. So the Munnu which is there, the second male gamete which is there, which was haploid in nature, fuses with the two polar nuclei. When I say two polar nuclei, haploid plus haploid, it is actually diploid in nature. It was actually diploid in nature before itself. Now when this diploid and this haploid comes together, N and N What will it bring out? 3N. 3N means what? It is going to be triploid. And what triploid? What is it forming? What is a triploid structure that is being formed? This triploid structure which is being formed is known as, first of all, what is the name of this cell? The name of the cell is primary endosperm cell. That means it's not known as central cell anymore, rather it is known as primary endosperm cell. Now, you will not call it as polar nuclei anymore. After the fusion, after this particular fusion has taken place, we'll call this nucleus as primary endosperm nucleus. Primary endosperm nucleus, PEN, right? PEN, pen is being formed. How is it? It is triploid, diploid, diploid. It is going to be triploid in nature. And since triple fusion is happening, why triple fusion? 3 nuclei are fusing. You will have a name for this, and that is known as triple fusion. Not understanding it? Listen to me carefully. First fusion happened over here. One male gamete said that female gamete, let's join together. Male and the female gamete join together. This is forming what? Diploid zygote. N, diploid zygote. This is plain reaction, and this is known as syngamy. Done. One more male gamete is there. That male gamete goes and fuses with the central cell. The central cell already had 2 nuclei. That means it was already diploid in nature. Now you go and give your haploid content also. So N and N and N, basically triple fusion is happening, which is why you will call this reaction as triple fusion. And what are you getting at the end of it? You're getting a cell which is known as primary endosperm cell. And what is the name of the new nucleus which is triploid in nature? It is known as primary endosperm nucleus, which is famously known as what? Pen. Which is famously known as PEN, that is pen. Please remember. Now, since 2-time fertilization is happening over here, I'll call this as double fertilization. One fertilization is known as syngamy, and another fertilization is known as triple fusion. Very, very, very important. So, since both syngamy and triple fusion occur in the same embryo sac, everything is happening, right? The process is known as double fertilization. This is unique feature of flowering plants. This is not seen in any other plant which is there. You know, angiosperm, gymnosperm, pteridophytes, bryophytes, lot of them are there. It is a unique feature of what? Angiosperms, that is the flowering plants. Now, what happens and why do you think this is happening over here? Why endosperm is being formed? This primary endosperm cell, no, later they will divide to form an endosperm tissue. But why do you think this is being formed? The primary endosperm cell, which later forms the endosperm tissue, the reason for it to be formed like that is that the endosperm tissue which is going to be formed later on will provide nourishment to the zygote, such that the zygote, which is a single cell, it's a single cell. Now you have to make it an embryo, right? You have to grow this up just like how a baby is being grown. Similarly, the zygote has to be transformed into an embryo, and for that you need nourishment. Who gives nourishment? Endosperm gives nourishment. So now you tell me, which of the cell is going to divide first? Does the zygote form the embryo first, or the primary endosperm cell, which is a single cell, is going to divide to form tissue first? It will be this one. Primary endosperm cell which is there, it divides to form an endosperm tissue first. Why? Because then only it can provide nourishment to the zygote out there, such that the zygote will be transforming into a embryo later on. You get it? And everything is happening inside what? Inside the ovule. Inside actually the embryo sac. Embryo sac is within the ovule. So we say it is happening within the ovule. And one more very important thing, when I say fertilization has taken place over here, when I say that over here in the embryo sac, your embryo is going to be formed, a zygote is basically formed, I'll now say it's not known as ovule anymore, it will be known as seed. It is going to be known as what? A seed. Once fertilization is done, you will not call it ovule, you will call this as seed. Got it? This is very, very important, and these type of things are something that you should not be forgetting. And I hope I had tried to explain you in slow manner, in a manner that you guys understand. You can rewatch this video as well if at all you feel that, oh my god, it's too much for me. Cool? Yes. Now, yes. So I have a question for you guys which can twist up your brain a little bit, but it's going to be good. And where is that question? This one. If a tetraploid plant contains 48 chromosomes in the nucleus, the number of chromosomes in the egg cell and the synergid respectively is what? Ma'am, what? Now what is tetraploid plant? What is tetraploid plant? Tetraploid means the ploidy of this particular plant is 4n. Tetra means 4n. It says that a 4n plant contains what? 48 number of chromosomes. Okay. That means the normal cells in the plant is 48. So normal number of chromosome in the plant is what? 48 chromosomes. Now, in its nucleus is basically a normal cell, only it is diploid. It is not something that has underwent meiosis yet. Okay, one cell which comes under this, that is a megaspore mother cell, that is the only one that undergoes meiosis. Rest everyone, they are just normal cells. Okay, the number of chromosomes in the egg and the synergid respectively is, they are asking about that. Listen. Talking about the ovule, here you have the cells of the nucleus. They have not underwent meiosis. They are just normal cells, and I am saying that all the cells have 48 number of chromosomes. Now, there was one cell, megaspore mother cell, which was for having 48 number of chromosomes, but from this cell I got the megaspore, right? From this cell I got the functional megaspore, only one of them, and from 48 it has reduced to what? 24 number of chromosomes in the megaspore because it underwent meiosis. Remember, it underwent meiosis. You got 4 megaspores, only one was functional. So we started off with 2n, and at the end you got n only, half. No. So if it was 48 over here, it will be half of 48. That means what? It will be 24 by the end, the megaspore. Now this megaspore forms the entire embryo sac. It forms the entire embryo sac. In the embryo sac, all the cells which we've heard about were haploid except the central cell. Now you have been asked about egg cell. Egg cell over here, it is going to be haploid only. It is going to be haploid. Half the number of chromosomes are going to be there, right? So over here, when I say it is haploid, that means that if it was 48 number of chromosome, the egg cell will have 24 number of chromosome. What about the synergids? It will also have 24 number of chromosome, only half of it. Okay, what about the other cells? Suppose you have been asked about antibodies. They will also have haploid number of chromosomes. That means they will also have 24 only. What about the central cell? Central cell, it will be different. Why? Because the central cell have 2 nuclei. One nuclei will be having 24. Another nuclei will also have 24. So it will be 48. Things are going to be different. If at all, it is what? The central cell. But here you have been not asked about central cell, you have been asked about the egg and the synergids. So always remember, okay, always remember, even though you say 2n becomes n, here instead of 2n you have been said that it is tetraploid. That means it was 4n, and now from 4n you do not have to make it n, you have to just make half of it. So if it was 4n, now it becomes what? 2n. All of them will be 2n except the central cell, which will be different, which is going to be different. Please know this. This is very, very important. So okay, so 4n, if at all 4n was 48, 2n will be what? 24, half of it. So egg and synergid will be having 24 number of chromosomes. So the answer for this question is going to be B. Got it? So this type of question can also come in KCET. I know it will be a little difficult for you to understand it. If you have doubts around it, please make sure you join the next class, which will be there on Thursday. I'll be coming on live on Thursday as well to take up the class. Works? Now, Aisha ma'am, please take the so the thing is that dear, for re-NET examination the timeline is very less, and you need to have more focused preparation, which is exactly why I am not taking it, because I might be doing the KCET related counseling videos. There can be distractions for me, and that's exactly why I have not taken up the charge for it. Do understand it. I really would love to teach you guys. It's not like I do not like to teach you. You can make the most use of the videos that has been already done, but I still request you to learn it from the teachers who are teaching for NEET itself. Okay, but if you have any doubts, I can be there to help you out. So dear children, with that we have ended up today's class. I hope it is understood. We have completed, as I told you, megasporogenesis and as well as double fertilization, and in between we studied about pollen pistil interaction as well. In the next session we will be talking about different type of pollination. Okay, and if I can, I'll try to cover up the next portion, that is post fertilization. So this event that were happening as of now, Okay, megasporogenesis or microsporogenesis, that was pre-fertilization event. Okay, then we saw fertilization, that is double fertilization. Now we will be also seeing about post-fertilization, what happens after fertilization. That is also something that will be there. Okay, so everyone who is watching this video will probably later on as well, do not forget to let me know how you feel this video was, and please take care of yourself. We are here to help you out. Okay, lots of love to everyone, and see you really soon. Bye bye.